The heritability of a trait determines its response to selection. That's why we're so interested in this heritability concept, which is really at the heart of our study of quantitative trait evolution. So in this lecture, we will dive into it, show what it is, how we estimate it, and how it predicts evolution under selection. The variance, remember, of some trait x is the average squared deviation of x from its mean, and that's just summarized here formally in a way you could calculate it. It's compact once you understand expectation notation, to just write this as the expectation or the expected value of the quantity x minus mean of x squared. Right? It's the average or expected value of the squared deviations of the individual observations. As we saw, the total phenotypic variance can be partitioned into components, the first split being that V sub p breaks into the genetic and environmental components, and then the genetic component is importantly further subdivided into the additive and other components. VD dominance variance is that contributed by interactions between alleles, where there's dominance and recessiveness at a locus, and then VI is the interaction between loci, so-called epistasis, which we won't go into because it tends not to be a very large contribution. There are actually two different forms of heritability. There's a broad sense heritability, which is in fact the fraction that's genetic in all its forms. That is to say, it's V sub g over the total phenotypic variance. It's usually written as capital H for heritability squared, which isn't the heritability squared. It's just the heritability. The square is there by convention to remind us that it's about variances, which are squared deviations. The narrow sense heritability is the one we care about for these purposes. It's the fraction that's additive genetic, hence little h squared is equal to V sub a, the additive component of the genetic variance, divided by the total. And remember, it's called additive because in this context, additive means adding up all the allelic effects over all the loci that affect the trait without regard to the combinations in which they occur. So that's the sense in which it's additive. And so, as we've said, the significance of little h squared is that it determines, first of all, the resemblance of offspring to their parents, which we'll see in just a sec, and second, the population's evolutionary response to selection, which we'll focus on in the second part of this lecture. So the easiest way and the quickest way to estimate little h squared is just to regress the phenotypes of offspring on those of their parents. And these figures tell that story. These are also from Heron and Freeman. It's probably hard for you all to see these very faint regression lines, but let me point that one out to help you. They go through, they're just the usual regression line that goes through a scatter plot. Here we just call this parents from short to tall, offspring from short to tall. If there's basically no consistent relationship, the heritability will be near zero. That is, knowing the parents' phenotypes does not tell us much about how tall or short the offspring will be. But if there's moderate heritability, let's say on the order of a half, then knowledge of the parents' phenotype, that is their height, will tell us a lot, but not everything, about the height of the offspring. It will explain about half of the variation in offspring height. And if heritability is really high, let's say approaching 1, then knowledge of the parent's phenotype, in this case height, lets us make an accurate prediction of the offspring's height. That is to say, the parents' heights explain statistically most of the variation in the offspring height. The definition of the regression coefficient, the slope in these kinds of plots, often abbreviated as B of y on x, is the covariance of x and y over the variance of x, that is, over the variance of the parental variable here. The covariance, you can look it up online or anywhere. It's a variance-like quantity that relates the covariation, that is the simultaneous variation, in bivariate observations, that is in points that have both an x value and a y value. It's the expected cross product of the difference in x from its mean times the difference of y from its mean. Anyway, if the covariance of x and y is 0, the variables are uncorrelated, like here on the left, because large values of y are just as likely to be associated with small values of x as with large values. But if the variables are strongly correlated and have the same mean and variance, then the covariance of x and y will be close to the variance of x, and hence the ratio will be close to 1. In this context, x is actually defined for outbreeding sexual organisms like us as the mid-parent value, that is the mean of the two parents' phenotypes, and y is the offspring value, the value of individual offspring. And we've already said this next point, the higher the slope, the better the offspring resemble their parents, which is to say, the better the offspring phenotypes are predicted by the parental phenotypes. Right, and that's what we just said. All right, so what is the heritability of height in humans? The textbook authors, John Heron and Scott Freeman, asked the students in their evolution course one year at UW to measure themselves, their siblings, and their parents. And they made one of these great data sets. Here's a graphical presentation of the scatter plot with mid-parent height in inches and offspring height also in inches. If you can see it, the regression, this very thin regression line, is quite steep. Its slope is 0.84, and that's the estimate of the heritability. So what fraction of the variance in height is additive genetic variance? It turns out it is 0.84. That was a clicker question. The right answer is that, and that's a fact you should simply burn into your memory. The heritability is the fraction of the total variance that consists of the additive genetic component. So that's then a very easy way to estimate the additive genetic variance. Just take it from the resemblance of the parent to the offspring. Now, a slight digression. In the latest edition of the textbook, Heron and Freeman define the y variables as mid-offspring heights, that is, as the average of all the siblings if there's more than one, just as mid-parent height is always defined as the average of mom and dad. That's actually incorrect. I was stunned when I discovered this in the 5th edition, contacted John, who I've known for years, and we had a nice email back and forth about it, and he admitted in the end that I was right. Here's the demonstration I made for the class last year, and for him, sent it to him to show that this is true. These are parent-offspring heights of the daughters only for our 36 Utah families in the genetic reference population. They have 1 to 8 daughters. There were 166 of them in all in the data set, which is an average of 4.6 per family. So here are the mid-parent heights of the 36 families that had daughters, in the data set on the x-axis, and above them the heights of all the individual daughters. The regression line passing through that has a slope of 0.82, very close to the estimate that Heron and Freeman got from their class. But if I averaged all the daughters in the families that had more than one, then I get a smaller number of points. Of course, there's just one per family, and they form a much tighter cluster. The slope of the line goes up to nearly 0.95, which is definitely an overestimate of the heritability. So its mid-offspring height is not the correct Y value. It's individual offspring heights. Okay. So back to what the heritability means and why we care about the resemblance of offspring and parents in this context. It's because it predicts the response to selection. And this was first studied, this set of ideas was developed in an agricultural breeding context in the early 20th century, where the people trying to improve the yield of corn, let's say, or whatever, would perform truncation selection. You have potential parents in your population. They show some normal distribution of some trait of economic value that you wish to, let's say, improve by moving the population to have a higher mean value of it. And so the traditional selection scheme is to pick a cutoff, alpha, let's call it, that is the bottom of the upper tail, let's say, of the distribution of individuals you will accept to be the breeders, the parents, for the next generation, and you won't breed from any of those with a parental phenotype of less than alpha. The difference of the mean, between the mean of this selected parent's tail of the distribution, shown by this vertical dotted line, and the overall mean of the entire parental generation pre-selection, is defined as the selection differential, conventionally denoted as capital S, big S, for selection differential. It's different from other little s's that we've used from time to time. Okay, then we breed from these parents, grow up the offspring, and measure their phenotypes, and what do we get? If our selection was successful, the mean of the offspring population, which is also a normal distribution of trait values, but the mean is above the mean of the parental population. The distance that we moved the mean, the improvement in the mean from the point of view of an agricultural breeder, is called big R for response to selection. So R for response, S for selection differential, or yes, selection differential is all we should call it. Okay, it turns out that the ratio of R to S, that is the fraction of the selection differential that we actually achieve as a movement or improvement in the population mean, that ratio or efficiency is another way to estimate the heritability. The response, it turns out, is expected to be the selection differential times the heritability, which is generally a number less than 1, meaning that we don't move the offspring population quite as far as we selected on the parents. And this, by the way, is why the slope of a line through a scatter plot is called a regression. You may have wondered that when you first heard it. What's regressive about it? You would have said to yourself. Well, it's called a regression because in Britain in those days, it was natural to say, the offspring mean or the offspring population regresses, that is, falls back toward the mean of the original parental population. So it was this regression or recession or whatever we might call it today that gave rise to the usage to refer to the slope as a regression. Okay, back to Heron and Freeman's hypothetical mice. Here's a selection experiment on tail length in mice. The population ranged from a little more than 8 centimeters to around 11 in their hypothetical world. They performed truncation selection by breeding from the mice with tails of 10 centimeters and up, colored red here on the distribution of mid-parent values. Then here are the offspring values, what they got back, not mid-offspring values, just offspring values. All right, they're the ones not selected are shown in blue, so all of the offspring that would have been produced if we had not selected are shown here, right, distinguished in color for actually selected from and then left aside, but this is what they would have been. And the point of this figure is to help you see that indeed the progress, the improvement, if you will, in the population that's achieved by the selection is defined by the slope of this line, by the rise over the run. That is to say, the slope in that sense does let us predict how much the mean of the offspring population will have been moved from what it would have been if there had been no selection. And so they show that here as the offspring, the original offspring mean, that difference between it and O star, the mean of the offspring from the selected parents, that is the red spots, and here's the selection differential for the parents, that is the difference between the original parental mean and the mean of the parents who were chosen as breeders. And there it is, right? The slope can just be defined as this ratio of these two offsets. The offspring movement generally smaller than the strength with which we selected on the parents, and there it is, right? It's literally R over S. Right, we've boxed those. Okay, so the relative change, the change relative to selection, the change achieved relative to the intensity of selection or the size of the selection, is the rise over the run in such a scatter plot. And so that's a way to remember how there are these two ways, different ways of estimating heritability. You can regress the offspring on the parents and shoot a regression slope through it, or you can do the selection experiment and see what evolutionary response you get. That's more complicated and time-consuming, but you could do it that way. They are, once you understand this graph, actually the same thing. So that means that the additive genetic variance, the part that creates the positive slope of the line, that is the resemblance of parents to offspring, is the part that makes the offspring resemble their parents. And that's why it plays such a critical role. Now let's take a break, and when we come back, put this to work, trying to help us understand how selection works in nature as opposed to in the selection barn at some agricultural institute.